Students, in this video we are going to start the second part of the chapter Reproduction in Lower and Higher Animals. In part 1 we studied about the definition of reproduction, sexual reproduction, how it occurs in animals. Regarding human reproduction, we studied the steps involved in the process of reproduction. Then we studied male reproductive system with the primary male organ or gonad, accessory ducts and glands. After the detailed study, we moved on to the female reproductive system. We studied the different parts of female reproductive system such as ovaries, oviduct, uterus, vagina, external genitalia, vestibular glands and mammary glands. After completion of this study of reproductive organs or system, we are moving on to the second part of this chapter, in which we are going to talk about the sexual maturity or puberty of human males and females. The age at which the reproductive system becomes functional, sex organs begin to produce gametes and sex hormones is called as puberty. In males, it is attained at the age of 12 to 15 years. Under influence of hormone testosterone, the secondary sexual characters appear, which we have studied in the last video. The voice becomes hoarse, facial hair appearing. Those are the secondary sexual characters. Thereafter, they remain functional throughout life. In females, the puberty onset is between 10 to 14 years, and it is characterized by onset of menstrual cycle, which is called as menarche, between 10 to 14 years. Mature females show cyclic changes in their reproductive system, which is a menstrual cycle. These cycles continue up to menopause, when a female becomes at the age of 40 to 50 years, she stops having the cycles, and which is known as menopause. Now, this period between the onset of the reproductive cycle and between the stop or end of the cycle, between menarche and menopause, this is called as the reproductive age of the female. Now, after this menopause, the female is not able to bear children, as she is not going to have the reproductive cycles. Menarche and menstrual cycle and menopause, these all are controlled by gonadotropic hormones, which are secreted by pituitary. Now, next we move on to menstrual cycle or ovarian cycle in female. Let us see how cyclic changes occur in females and how she is able to produce the female gamete. It is a characteristic feature of primates, including human. Involves series of cyclic changes in ovary and female reproductive tract, mainly in uterus. Now, this slide has a diagram which will show the cyclic changes that are occurring during the menstrual cycle. Now, here you can see this is the wall of uterus, and here the arrow is pointed. Here the changes in the wall of uterus are shown. Now, it has a periodicity of approximately 28 days. Middle of each cycle has the period where the egg or female gamete is released. This egg in every cycle comes alternately from one of the two ovaries. Now, if left ovary produces a female gamete in one cycle, then in other cycle the right ovary will produce the gamete. The cycle is divided into 4 phases. Now, we are going to study those. First is menstrual phase. The beginning of each cycle is the first day of menses, or where the blood loss is started, which is approximately 45 to 100 ml. It lasts for approximately 5 days, average 3 to 7 days. Endometrium of uterus. Now, I am going to show you again the previous diagram. This region is called as the endometrium of uterus. Now here this endometrium is sloughed off, or it ruptures, and in form of mucus blood it is released, and which is called as the menses. Endometrium of uterus breaks down under the effect of prostaglandins released due to decreased level of progesterone and estrogen. Now all this cycle is a delicate balance of hormones. Decreased level of progesterone stimulate and estrogen stimulates prostaglandins, and the uterine wall is locked off. Blood, tissue fluid, mucus, endometrial lining, and unfertilized oocyte is also discharged. That is why the menses or menstrual phase indicates that there is no pregnancy, or there was no fertilization of male and female gamete. Unfertilized oocyte also is released along with this blood and tissue fluid. Due to the blood loss and loss of this endometrial thickened wall, endometrial lining becomes very thin, about 1 millimeter. Menstrual discharge continues for an average of 5 days. This blood does not clot due to presence of fibrinolysin. Menstrual phase occurs when ovulated egg does not get fertilized. It is called as funeral of unfertilized egg. Now the next phase is during these 5 days during menstruation, many primordial follicles developed into primary and few of them into secondary follicles under the effect of FSH. Now mind you, when there is no fertilization, again the ovary prepares itself for the production of next female gamete. So development of primary or secondary follicle again progresses, and now it is going to form mature or Graafian follicle. Next is proliferative phase, follicular phase, or postmenstrual phase. Now during proliferative phase, again what happens is, after the end of menstruation, there is between 15 to 30 days secondary follicles proceed to develop, but usually one of them develops into a Graafian follicle. Then while another secondary follicles degenerate, they undergo atresia. These secondary follicles secrete hormone estrogen. Now during menstrual phase, it was indicative of there was no fertilization, and the earlier follicle or oocyte that was developed is wasted, or it is given out, it is released outside body with the menstrual flow. Now it is time to develop another follicle for fertilization. So the developing secondary follicles secrete hormone estrogen. Now this is responsible for development of follicle into mature follicles, and this proliferation of new follicles is under influence of gonadotropic releasing hormones, which really stimulates release of FSH. Now this is all orchestra, endocrine orchestra. Hypothalamus stimulates pituitary, and pituitary stimulates the ovary for secretion of hormones. Endometrium now in this phase begins to regenerate under effect of gradually increasing quantity of estrogen. Now during this phase, why this is called as a proliferative phase, again endometrium wall starts to thicken. Now I'll again go to previous slides so that you will understand. See here, during this proliferative phase, the loss of endometrial wall is again regenerated. Now the endometrium starts regenerating. Now you can see gradually the endometrial walls starts thickening, and that is why it is called as a proliferative phase. This phase is under the influence of hormone estrogen. New proliferation of new follicles is under influence of GnRH, which stimulates FSH. Now here you can see the levels of hormones increasing. Now in proliferative phase, estrogen is increasing, progesterone is very low. Then endometrial thickness gradually is increasing and it reaches up to 3 to 5 millimeters. Then next is ovulatory phase. At the mid of the menstrual cycle, the mature or Graafian follicle undergoes ovulation, releasing ovum. Now here it is shown. Changes in follicle are also shown here. In proliferative phase, secondary follicle starts maturing to Graafian follicle, and at the 14th day there is ovulation, release of ovum by the ovary. Endometrial wall starts to thicken between 3 to 5 millimeters. Ovulatory phase is characterized by release of ovum. Graafian follicle releases ovum or secondary oocyte into the pelvic cavity, and that is the 14th day of menstrual cycle. Rapid secretion of LH by positive feedback mechanism causes rupture of mature follicle. Now this rupture is due to luteotropic hormone. Ovulation may be in ovulation there might be mild or severe pain in abdominal cavity. Then last is secretory phase, which is between 14th to 28th day of cycle. 14th is the ovulatory phase. 15 to 28 is the secretory phase. Now you can see here, this is the secretory phase. Now longest phase, it is between 15th to 20th day after release of secondary oocyte. That is the previous phase of it, ovulatory phase. When there is secondary oocyte released, remaining tissue of Graafian follicle is transformed into a scar-like tissue called as corpus luteum under effect of LH. Now this is corpus luteum. Then corpus luteum begins to secrete progesterone and estrogen. Now you can see levels of progesterone have increased here. Small amount of estrogen is also secreted. If ovulated egg gets fertilized within 24 hours, then this corpus luteum maintains the pregnancy. But if there is no fertilization, then corpus luteum in 2 weeks degenerate into white scar, which is called as corpus albicans. Okay. After ovulation, the Graafian follicle transformed into a scar-like tissue, which is called as corpus luteum. Corpus luteum secretes progesterone, that is why the levels have increased. But if there is no fertilization, this corpus luteum degenerates to form corpus albicans. Under influence of hormone progesterone, small amount of estrogen and inhibin is released by corpus luteum. Endometrial glands grow and they become coiled and start uterine secretions. Now you can see here the endometrial wall thickness has become greater and the glands in the endometrial wall have become coiled and they are going to secrete uterine secretion. Endometrium has more blood supply, thickens up to 8 to 10 millimeters. Inhibin now stops secretion of FSH. follicle stimulating hormone now is minimal. These changes are necessary for fertilization and subsequent implantation. Now, you might wonder why there is such a thickness of endometrial wall, because if the ovum gets fertilized, now it will pass through the fallopian tube and it will enter the uterus, and the embryo now will get implanted into this uterine wall. That is why the thickness, this is actually the preparation for the implantation of embryo in the uterine wall. If the ovulated egg gets fertilized and embryo is implanted, secretion of human chorionic gonadotropin. Now this is very interesting. If there is pregnancy, that means the embryo gets implanted, there is one more hormone that comes into play, which is called as chorionic gonadotropin, HCG, human chorionic gonadotropin, which extends the life of corpus luteum and stimulates its secretory activity. Now remember, this corpus luteum has activity of secretion of progesterone, which is ultimately responsible for maintaining pregnancy. Now, HCG, secreted after implantation of embryo, now it increases the life of corpus luteum. Presence of HCG in maternal blood and urine is an indicator of pregnancy. So this is the hormone that is very crucial, and it is identified by the pregnancy kit which is available in the market. And in absence of fertilization, now the next menstrual cycle begins. Now if there is no fertilization, all this thickened endometrium again will degenerate and menses will start again, starting the menstrual cycle, menses of the menstrual cycle. Then we go to the next slide. This is all the description of the different phases of menstrual cycle, which you can refer later for understanding. Here is the secretory phase, which is the longest phase. Now gametogenesis, this is very important part here. Gametogenesis is the process of formation of gametes in sexually reproducing animals. The male gamete is sperm and female gamete is ovum or egg. It is formed from the primordial germ cells of gonads. Now gonads are testes in male and ovaries in female. Now there are cells called as primordial cells which give rise to these gametes, and let us see how that happens. Now we will first start with spermatogenesis. Spermatogenesis is process of formation of male gamete or sperm or spermatozoa from germinal epithelium of testes. Now in previous video we had studied the structure of testes. Testes has different covering: tunica albuginea, tunica vaginalis, etc. And if you take a section of testes, you can see many small compartments and having tubules called as seminiferous tubules, which show the development of spermatozoa. Now at the onset of puberty, hypothalamus, now you can always see that hypothalamus is the first one in the endocrine orchestra. Hypothalamus stimulates pituitary, and pituitary ultimately stimulates gonads. Hypothalamus begins secretion of gonadotropin releasing hormone GnRH. GnRH initiates the significant increase in follicle stimulating hormone, which induces spermatogenesis. Follicle stimulating hormone, name itself suggests that in males it is promoting spermatogenesis and in females it is promoting oogenesis. Now each seminiferous tubule is lined by a single layer of cuboidal epithelial cells called as germinal epithelium. In previous videos we have seen in transverse section of testes you can see many spherical seminiferous tubules, and at the periphery of these seminiferous tubules you can see cuboidal cells, which are the cells giving rise to the different stages during spermatogenesis, and those cells are called as germinal cells of germinal epithelium. Cells of germinal epithelium undergo spermatogenesis to produce sperm. The process of spermatogenesis involves three phases. First is multiplication phase. Now these primordial cells, they undergo mitosis and they produce large number of spermatogonia. Now mitosis is equational division. It is just increasing the number. It is just multiplying the cells. Now when the spermatogonia are formed, then they enter into a second phase called as growth phase. Spermatogonium, which is diploid, 2n, 46 chromosomes are there, it stops dividing, grows in size and develops into primary spermatocytes. Now here you can see spermatogonium, one of them has grown into primary spermatocyte due to accumulation of food material. Now this is called as a growth phase. Primary spermatocyte is again diploid only. Then maturation phase. Now this is very interesting to understand. Maturation phase involves initial mitotic division and then meiotic division and then spermiogenesis. Now here where the meiotic division comes into play. After meiosis, haploid gamete is going to get formed. Now meiotic or reduction division is the part of this maturation phase. The spermatocyte undergoes meiosis 1, forming secondary spermatocytes. Now you can see here primary spermatocyte has entered meiosis 1 and formed secondary spermatocyte, and which is now a haploid. Then secondary spermatocyte undergoes second phase meiosis 2 to produce 4 haploid spermatids. Now you can all see that these are spherical circular cells. These cells now, in order to function as male gametes, motile moving gametes, they have to undergo transformation, and that is called as spermiogenesis. Spermatids now have to show modification in their shape and structure so as to work as a motile moving gamete, which has to move towards the female gamete and show fertilization. Now this phase is called as spermiogenesis. From spermatid, formation of sperm takes place. Non-motile, non-functional spermatid gets transformed into a functional spermatozoa by a process called as spermiogenesis. Spermatid remain held to each other and to Sertoli cells. Now here you can see the junctions between the spermatids and the Sertoli supportive cells is very tight, and they have cytoplasmic bridges. The sperm heads are attached to Sertoli cells and the tails hanging in the lumen. Now here heads are attached to this nutritive cell and the tails are present in the lumen of seminiferous tubule. Then during spermiogenesis, the length of spermatid increases. Centrioles are rearranged as primary and distal centrioles. Now here at the neck region, you can see the centrioles are present. Then mitochondria become spirally coiled and acrosome is formed in the Golgi body. Now The mitochondria in spermatid, now they become spirally arranged and they form the middle piece, and the sorry centrioles are present in the neck region. How is the structure of sperm? Sperm has a head, oval in shape. Haploid nucleus is present in the head. Above nucleus is cap-like structure called as acrosome, formed from the Golgi body present. Acrosome has hydrolytic enzymes, hyaluronidase, and proteolytic enzymes like zonalysins and corona penetrating enzymes. Now, when the sperm approaches the female gamete, it has to penetrate through the layer surrounding the gamete, female gamete, or the ovum, secondary oocyte. So that is why the acrosome contains the different lytic enzymes which will help the penetration of sperm inside the ova. Neck is a very short region having two centrioles, primary, proximal, and distal centriole. Now you will see here centrioles are present in this region. Then the next region is middle piece. It has axial filament surrounded by 10 to 14 spiral turns of mitochondria, which is called as Nebenkorn. This is very essential because this provides energy for movement of sperm. Sperm has to move, it has to show the movement through the female genital tract. So this middle piece has a spiral of mitochondria providing the energy. Tail is a long, slender, tapering part, has cytoplasm and thread-like axial filament. Passing through this middle region, there is this axial filament, and surrounding it is a cytoplasm. And at the tip of the sperm, at the tip of the sperm, there is the end piece which doesn't have plasma membrane. Only the axial filament can be seen. So this is how the male gametes look like. Now the next part of the video is oogenesis, formation of haploid female gamete, egg or ovum from the diploid germinal epithelium. Now this oogenesis is very interesting. It has the same phases like the gametogenesis in males, multiplication, growth phase, and maturation phase. But it is a little discontinuous. This process is halted at certain time points and it is not a continuous process. This takes place in ovaries, involves process of meiosis and mitosis, and like spermatogenesis, it has multiplication, growth, and maturation phase. Now let us start with multiplication phase. The primary germinal cells, primordial germ cells, which are diploid, present in ovary, undergo repeated mitotic divisions, which is similar as the male reproductive system, to form millions of gamete mother cells or oogonial cells. There it was spermatogonial cells, here they are oogonial cells. The process is completed in the embryonic stage of human females. Now look where it starts. It starts way before the female child is born. Then the growth phase has some of the oogonia stopping division and begin to increase in size and forming primary oocyte, which are again diploid. Cellular organelles like ER, GC, and mitochondria increase in number. During growth phase, they stop division and they increase in size. Third is the maturation phase. The process is initiated prior to birth in female baby. Primary oocytes. Now, first let us see from primordial germ cells. Due to repeated mitosis, oogonial cells are formed. Oogonial cells stop division. Some of the oogonial cells stop division and form primary oocytes which are diploid. Now these primary oocytes undergo maturation phase, which includes meiotic division 1 and 2. The primary oocytes undergo meiosis 1 to form two haploid daughter cells. This division is very interesting because the cells, two cells, daughter cells, which are formed, they are not of same size. One is larger and other is very smaller. The larger one is secondary oocyte, which is haploid, and another small cell is called as a first polar body. Secondary oocyte precedes meiosis 2 up to metaphase 2. Now, interestingly, now here also, this process is stopped at meiosis 2 of, metaphase 2 of meiosis 2. The secondary oocyte is shed from Graafian follicle of ovary. Now remember, the secondary oocyte is arrested at a stage metaphase 2. Now this restarts and completes meiosis 2 when and only when the sperm fertilizes it. This last phase is usually completed in the ampulla of fallopian tube at the time of fertilization, where when from ovaries the ovum is released, it is picked up by the fimbriae of fallopian tube, and sperms which are released in the female genital tract, they travel through cervix and uterus and they reach the ampulla of the fallopian tube. So you can say that this process is not continuous, but it has breaks in it. Then in this division also two unequal daughter cells. Now the secondary oocyte arrested at metaphase 2 is fertilized by a sperm, but again there are two unequal daughter cells formed. The larger cell is ovum haploid and the smaller is the second polar body. Ovum form functions as female gamete and ready for fertilization. Completion of meiosis 2 and completion of fertilization goes hand in hand. Now, if the secondary oocyte does not receive a spermatozoa, it is shed along with the menstruum. So, when we studied the menstrual cycle, the first stage, menstrual stage, at this stage, if there is no fertilization along with blood and endometrium, the secondary oocyte is also released. Now here is the structure of secondary oocyte. This secondary oocyte is surrounded by a lot of cells. These cells are called as corona radiata cells. Now here you can, I'll just point out so that you can see. These are the corona radiata cells. Now here you can see the first polar body. Actually these both are divided from the oogonial cells, but they due to unequal division, the secondary oocyte, from primary oocyte, the secondary oocyte and the first polar body is formed. Now here this region, the large nucleus, is called as germinal vesicle. In humans, the released unfertilized egg is called as the secondary oocyte. Non-cladoid, it is without shell. Like in aves, you can see they are shelled. They are having the shell made up of calcium carbonate. Then microlacethal, very less amount of yolk is present. In hen's egg, you can see a ball of yellow yolk present, but here in human egg there is very less yolk present, approximately 0.1 millimeter or 100 microns in size, rounded non-motile haploid female gamete. Nucleus of the egg appears large, and this is called as a germinal vesicle. This is a germinal vesicle. Typical nucleus or pronucleus is formed at the time of fertilization. Cytoplasm of egg, also called as ooplasm, which has no centrioles. Egg is surrounded by various coverings. Now this is interesting. The egg membrane is vitelline membrane. It secretes zona pellucida, a non-cellular glycoproteinous membrane secreted by vitelline membrane outside the vitelline membrane. The inner is vitelline and outside to it is zona pellucida. Corona radiata is layer formed by several radially elongated cells adhering to one outer surface of zona pellucida. These cells are formed from the innermost layer of granulosa cells firmly held to zona pellucida and to each other by hyaluronic acid, which is a mucopolysaccharide. Now you can see these cells are kind of radiating, so that is why they are called as corona radiata. They are attached to each other and to the zona pellucida layer very firmly. Between vitelline membrane and zona pellucida, there is a fluid-filled cavity called as perivitelline space, and first polar body lies in there. Now the inner is the vitelline membrane and outer to it is zona pellucida. Now the space between is called as the perivitelline space. Now the first polar body is present in there. Egg shows polarity. The side having germinal vesicle and first polar body is called as animal pole. This is the animal pole and opposite to it is vegetal pole. So this is the structure of secondary oocyte. Here we complete the gametogenesis of human males and females.